Okay, so we will continue to talk about the history of the microorganism. So this is something really important. So we talk about microorganism. And we already talked about in our first lecture, it is too small to see by naked eye or aided eyes. So therefore we need a tool. That tool we call the microscope. And some of you in the first lab, we already practiced using the bright field microscope to do the observation of emitted slides. So lots of you will be practiced today and the following couple of weeks. So who is the first person to really create the microscope? His name is Anthony van Leeuwenhoek. Now, when you see anybody, the middle name has a van, that means this person most likely is from Dutch, Netherlands, and or Belgian area. So it's true, he's coming from Netherlands. So we call it a Dutch scientist, and he called himself very humble, is a Dutch amateur scientist. Because his major work is doing the business. He's selling the clothes, selling the other things. He's doing the business. His hobby is to do some of the scientific stuff like micro stuff. So when you see here, you can see he's a mechanic microscope. This mechanic microscope and this guy, the magnification is not really strong. It is about 200. Some literature says it's about 50 to 200. Because it's quite simple. When you see that, you see it has a hold, it has a lens. That's it. And of course, there is something like a stage the light will be passing through. So what he found? He found these bacteria. But what are they? Later on, people say these type of things are not really the morphology of the bacteria. That means the bacteria moving, so it's a bacterial flagella. That's later on, like 300 years later, 200 years later, people go back to see the history literature review. They find these are bacterial flagella, which means the bacterial movements. However, he really is the first person to well describe the microscope and describe the bacteria. The reason is he published a paper. So that's why in the lab I ask you guys to do a good job of recording, because when you recorded something and put your signature, put your name there, which means that's copyrights and the property is belonging to you. That's what he did different from the others. Lots of people in that time is claimed that they find the microscope, they do the observation of microorganisms, but they forgot to record it. But they also have some bad things for him. This is 1623 to 1723. He had a very long life. Look at that. That's almost like 90 years old, 80 years old. When we talk about Louis Pasteur, this is 1822. We go Bob Koch, that is almost 1910. How about that period of time? There is almost 150 years in the gap, is that right? Why? Because one of the things he didn't do very good, he's a businessman, and he refused to sell his microscope to the scientific area. We don't know the reason behind it. Maybe people don't pay too much. He refused to sell his magnific microscope to the other person, which is delay the development of the modern microbiology area for about 150 years. Because here is the thing, why the microbiology area can develop? There are three major things we need to know. Number one is we need an electron microscope to see the morphology. This is the shape of the bacteria. That's the first thing. The second thing, we need biochemistry, biochemical, and the physical, and the physiology of bacteria. Last one is molecular stuff. Is molecular biotechnology. Those are the three things to push the development of the microorganism and the microbiology. That time, if this is delayed, then everything else will be delayed. Okay, so that's something for Anthony van Leeuwenhoek. Okay, the next thing what we're going to talk about is spontaneous generation. That is a very interesting topic, and even today, people still debate about it. Some of them are scientific reason, some of them are religion, or maybe politics reason. People debate about that. So, what is spontaneous generation? Spontaneous generation, that means you have a non-live subject, and which will be automatically generate a live body. So I'll give you a very easy example. Let's say we're going to talk about later on. We have a meat. The meat will be automatically generate a fly. Now, by the time 300 years ago, people believe it. Because when you put a meat on the bench, on the desk, for about like a week, maybe not even a week, like 2 days, 3 days, if it's a very high temperature, you see the fly comes out. And then you see some eggs there even. Then you say, okay, that's a meat that generates the fly. So that is spontaneous generation. So the scientific area had a big debate by the time. Now, who is the first person really discredit this spontaneous generation theory? Is Francesco Redi. This is an Italian scientist, and he did a very famous and very important research to discredit spontaneous generation, which is right on the slides there. So what is the story? If we say very simple, I have a meat. I have a chunk of meat, okay? Then, if I expose to the air, this meat later on will see something. Some like an egg there. Not really egg. We call it an egg. We call it a maggot. Sometimes we call it a larvae, we call it. Then what comes out? A fly that comes out. Okay, that means meat generate a fly. Yes, it's correct for spontaneous generation. So we call this is like a control. It's a positive control. Then he did another experiment. Okay, I'm going to be cover the meat. This is the meat. I'm going to cover the meat with a paper. Had the paper covered. A solid paper covered. So what's going to happen? There is nothing going to generate for the fly. Okay, what this tells us? Which means the meat will not generate the fly. So, this theory is wrong. The spontaneous theory is wrong. Then we're going to find out where this fly that comes out. So, it's very magic. In a third test, he had a meat. He did is a cheesecloth. We call it a cheesecloth. You do it. If you have a bleeding on your hand, sometimes put a cheesecloth, and then will be stopped bleeding. So he found a cheesecloth. We call it a cheesecloth. Now, what's the magic for these cheesecloths? There's a space between that. Is that right? There's a space. So, what happened? The maggot, the egg could be there, and then the fly comes out. So this is obviously a very smart method to tell us The fly is not coming from the meat. The fly is coming from the fly in the air because it created the maggot, the egg, and the egg later on generate a new fly. However, this test, did you see what they're missing? What are the two things will generate a fly? Number one is the meat, is that right? Number two is what? Is the air. In this experiment, you cannot avoid the air. You cannot tell us the air is not generated fly. I can debate it. I said it's air generated. Is that right? Because the air is still there if you have a cheese cross. So, but by the time, this is a very good experiment, and from the, we will say from the non-microorganism level, this is discredited spontaneous generation. But having solved the problem is how are you going to verify the air is not generate a live body? This is what later on they do. Okay, so once this experiment finished, then people move on to the microbiology area. They say, is spontaneous generation will be true for microorganism? Okay, then they do the two experiments. Obviously, there is the two results you're going to come out. Let's say I have a flask. Okay, I have a flask. Let's say this is a beef extract liquid. Something like you put a beef stew there. Okay, you put a beef there. Beef stew, then you boil it or you heat it, just put some water. Okay, let's say if you boil it. If you're boiling, it is around 100 degrees Celsius, not really. In here, maybe 95-ish. Okay, 100 degrees Celsius. It will generate two results. One result is no bacteria. Now, of course, after boiling, you're going to cool down a little bit. Okay, stay in the room temperature for about 5 days or 2 days. No bacteria. Very clear. However, some people find another result's going to come out. This becomes very messy, very turbidity. Or we say very cloudy. Which means there's a bacteria there. Or microorganisms there. So, which one is correct? Now, these days we know that's all kind of thing could happen. The bacteria could be heat resistant. The bacteria could have a spore, could have an endospore. Is that right? But these old time people doesn't know. So, John Needman, which is a British scientist, he did the experiment. The mutton broth in flask, boiled, sealed, he finds it's cloudy and it contains microorganisms like that. Then the Italian scientist, Lazzaro Spalletti, he finds his experiment, if you, in the beef extract, you boiled, then no bacteria. You sealed, no bacteria. Now, of course, these have to be sealed. Okay? Okay, how do we know it is? How? Because people can debate. Oh, it didn't generate the bacteria. But here, these mutton broths or beef extract broths generate the bacteria, even you heat it. Because later on, it's created a bacteria. So that means the spontaneous generation in the microbiology level is not discredited. It's highly possible. 50% chance. So who is the person who did that? Louis Pasteur. In 19th century, he started to do the experiment. This experiment is really magic by the time. So what happened? Instead of use these very traditional flasks, okay, it's the same thing. He created this very magic stuff right here. This is called a swan neck. Like a swan, like a goose, like a neck. Okay, so what did he do? Once this experiment he has a broth, okay? The broth, we're going to boil it. be boring. 100 degrees Celsius, let's say for 2 hours. What do you do? You're going to broke it. So it's one set of the test is he broke it. So what happened? It's become a very traditional flask like this. Okay. Then what you see? You see a lot of bacteria there. The bacteria grow. So where they come from? Why the bacteria grow? Because the bacteria from the air goes in. Now he has another set. What's the second set? This is the one, he did not break the swan neck. He goes here, he goes here. If you keep the swan neck, there's no bacteria. This is pretty clear. Because the edge here, the bended area, which is hiding bacteria. And the bacteria cannot come in, therefore, it's not. So, what this means? Which means this broth not automatically generate bacteria. The bacteria is coming from the air, they are coming. Why the broth becomes turbidity? The reason is there's a bacteria there. So, this is, if you read, you can be very clear to see this spontaneous generation, which is discredited in the microbiology level, and which means the bacteria is coming from the air, from the broth, is not automatically generated from the broth. Now let's go back to here. How come there is a two different type of the results generated? Especially, even you boiled, you still see the turbidity of the bacteria. Then that's a 2% contribution. John Tydell, which is a great British scientist, he found that some of the bacteria will be resistant to the heat. So we call it a heat-resistant bacteria. And I tell you one thing, they do have a heat-resistant bacteria. Where are they? Did you ever go Yellowstone in the hot spring? You see the hot spring is 120 degrees Celsius sometimes. Why the bacteria can survive there? Heat-resistant bacteria. One of the bacteria we will do in the lab, we will mention, which is also heat-resistant, is bacillus, especially geobacillus. So we do have these type of the bacteria, which is heat-resistant. Then we need to know why some of the bacteria is heat-resistant. Then Ferdinand Kohn, another great scientist in England, he found that lots of the bacteria could be heat-resistant because of the bacteria has endospore. And bacillus is one of the group of the bacteria naturally has endospore. Another one is Clostridium. Most of you from West Virginia, you know the West Virginia area, they do the canned food. Is that right? You put the pickles, tomatoes, everything is there canned. What's the regulation for that? So regulation right now for the Department of Agriculture is to make sure the pH is lower than 4.0. Because that lower pH will highly prevent the endospore to generate. And those type of the food, we call it a low acid food. It's a very tricky stuff. Will cause endospore. So that's endospore. Bacillus is one of the bacteria has endospore. So Ferdinand Kohn find the reason for heat-resistant bacteria is they can generate endospore. And we will do the endospore stain in the lab next week. So you will see an endospore, what it is looks like. Okay? Okay, next great person, Bob Koch, a German scientist. He did a big contribution for this area. Today, we are doing lots of the work in the lab, like agar, like median, all those things is all coming from his group. So we will mention those one by one. And by the time I also let you know, he and Louis Pasteur are not friends. They are big competitors. Louis Pasteur is from French and he's from German, big competitor. People memorize Louis Pasteur, had a Louis Pasteur microbial institution in Paris. Then they started to have that branch institution in like China, India, South America, all kinds of the countries. Because they want to memorize him. But I tell you, by the time, they are big competitor. Now, Bob Cook, the most important thing, what he did, is this Cook postulate. This is well defined, the relationship between a bacteria, a microorganism, and a live body, which is an animal or a human being. So Bob Cook have a postulate. So what is that? So first thing, which means a healthy animal absence of certain microorganism. And sick animal present with certain microorganism causing the issue or cause the sickness. So, very simple example. If a guinea pig or a human being, let's say a pig, has tuberculosis, which is right there. Tuberculosis. Then, of course, you will be isolated. Mycobacteria, tuberculosis, from the lung area of this pig. You could isolate it. And he pointed, if it's a healthy people or a healthy pig, you should not be finding microbacterial tuberculosis in the pig, which means absence. Okay, this is the first one. Second, if the pig has microbacterial tuberculosis, the microbacterial tuberculosis can be isolated from the pig and get a pure culture. So what that means? Which means a microorganism can be isolated from sick animal and generate a pure culture. We will talk about this pure culture later on. Very quick, very interesting topic. What is a pure culture? This is the second one, which means you can isolate and generate a pure culture. Now, what's the third one? The pure culture, if you contaminated it or infected it, I usually call it contaminated. Medical area, they call it infected. A healthy animal, this animal will get same disease. So what that means, if there is this microbacterial tuberculosis coming from a guinea pig, if this microbacteria going from let's say swine or a rabbit, then they will get the same disease, which is they also going to get tuberculosis. Now, last thing. This microbacterial tuberculosis can re-isolate it from swine and the rabbit. So, I would just say, re-isolated from sick animals. These are 4 major points for this Koch postulation. Well describe the relationship between a microorganism and a live body. But I want to tell you one thing, even today, you know there is something behind it. It's not really 100% true. Are you really sure the sick animal is absent of these microorganisms? Not really. What's missing here? We have asymptomatic people for coronavirus, is that right? You still get isolated. Why? Immune system, is that right? The immune system will play the role there. But by the time they only have a microbiology, they didn't have an immunology yet. Later on, maybe 50 years later, the people start to talk about immunology because they find the Burson fabricus, which is the butter area of the chicken, has a B cell. We will talk about that later on. So usually microorganism and immunology we talk right now is combined. There are some classes in the medical school, we call it a microbiology and immunology class. That's the reason. Because there is no immunology concept right here. Okay? So that I want to tell you one thing. This is some tricky here. Now last one. Did we really can re-isolate it? It depends. Did you ever hear about typhi Mary, Salmonella typhi? Mary Mellon, he had Salmonella typhi. However, he never had a problem. And he very difficult to get isolated. When she died after about 50 years, in 1983, Stanford University isolated the Salmonella typhi from her blood area. Because it's hiding there, which is packaged by a macrophage and protected. So the re-isolated sometimes is very difficult. It does not mean the sick animal, you infected, you will be re-isolated with the same bacteria. It's also the tricky there. And also, some of the microorganism, it is viable but non-culturable, which means you cannot use traditional methods to grow them. You can only rely on the modern molecular microbiology to test the sequencing of that. So that is why if you're testing coronavirus, you usually do the PCR test. Is that right? Because it's difficult to grow sometimes. It takes a longer time to grow. So that's why I wrote there the morphology, the biochemistry, physiology, and also the modern molecular biotechnology, the 3 things to prompt the development of the microorganism. Okay, microbiology area. So this is a Koch postulate. Okay, we mentioned about the pure culture. Then we're going to talk about that in next couple of slides. So these slides and these slides, all these we usually put them together to talk about that. So we're going to talk about the topic is pure culture. Okay, what is pure culture? Pure culture, if you want to say it very simple, it is a group of bacterial cells coming from the same mother cell. Now, how to get a pure culture? In the history, spending many years to develop it. It's related to lots of the information behind it. There is a 2 group of the research group in the world is doing all kinds of research by the time. Who are they? I just mentioned. Is the Louis Pasteur group and the Bob Koch. They have the #1 debate. When you get a pure culture, is that should be done in a liquid media or has to be done on a solid media? Louis Pasteur group think about has to do, we can do this in a liquid media. So what is liquid media? Which means broth. So how to do it? He thinks it can be done. Okay, I have a cube of the bacteria. Let's say I have an E. coli. Okay, how many cells there? 10 to the 8th cells per ml. I want to get a pure culture. What we do? I'm doing a serious dilution. This is, some of you like it, some of you don't like it, like you do in the chemistry class. Okay, I prepare so many tubes, on and on. Each of them is 9.0 ml. And I'm going to add 1 ml into the first one. Then mix them very well. Then you add 1.0 ml, go to the second one. Then you got to do this all one by one. This is called tenfold serial dilution. Okay, if that works, theoretically yes. When I do this on and on, I will find one cell. But there's a problem. How are we going to verify that? The last tube is only 1 cell. Is that right? Okay, then Bob Koch's group said, this is wrong. You can't do it. We have to do is using a solid medium. Okay, if it is a solid medium, then we have another question. What type of the material can make this solid medium? They're spending a longer time to think about it. Let's say they tried what? They tried jelly. It didn't work very well because the jelly later on broke. What's the tricky things here? The bacteria need to growing, but not eating them. That's challenge number 1. Challenge number 2. This media has to be melted at higher temperature, but it has to be solidified in a low temperature. So they say melted at high temperature and solidified at low temperature. So you need to find those materials kind of very tricky. Okay, they're spending a lot of time. They try to do it. There is one person in Bob Koch's group, which is Hesse. The last name is Hesse. Let me make sure the spell is good. H-E-S-S-H-S-E, which is Bob Hesse. Bob Hesse, underneath Bob Koch's group, spending 3 years to find the material. There is one very summer time and a very hot time in summer in Germany. His wife, Fanny Hesse, prepared the lunch because he's always working and he like a housewife and just prepare the lunch. He keeps thinking, in the very hot summer Germany, it's about like 35 degrees Celsius, probably like 90 degrees Fahrenheit-ish around, okay? One day she said, honey, it's for lunch. Can you just take a break? Then Bob Hesse says, what do we for the lunch? Fanny has said, we're going to do pudding. French pudding, okay? That you like it. Then Bob Hesse think about pudding. That's a good material because even at 35 degrees Celsius, it's still solidified. Is that right? So he tried that in the lab, and the bacteria grow there. And they grow a very good colony. We talk about this, the pure culture. And what's the material for pudding? It's agar. Is that right? So they did lots of the repeat of the experiment, the different bacteria. They finally find using agar we will generate a solid media. The solid media can get isolation of the pure culture. Now, what is that? We will practice this in the lab a lot. This is the agar plate. Okay? And I have a culture there. This is a broth, let's say. The bacteria is there. How are we going to get a pure culture? How are we going to do? We do a streak plating. How to do a streak plating? You pick some loop, pick some bacteria from there. Very intensive at the first quarter. Then you flame the loop to do a second one right here, less intensive. Then you flame again to do the third one right here. Then later on, you're going to do the last one. It's like this. And after 35 degrees Celsius, 24-hour incubation, you will see there is lots of the colony, lots of the dots will generate. And this is a single bacteria colony. This representing a pure culture. But be careful. This bacterial colony is not one cell. It is not one bacteria cell. Since it's called colony, which means a group of the bacterial cell attached, accumulated together, how many population are there? At least 10 to the 6 cells per ml. That's one colony. Now, what means a pure culture? You know E. coli? Escherichia coli? That's a pure culture. Because only one genome name and one species name. Okay? So, this is a story. Now, when you have agar, people are going to ask you, I need a container to put the agar, is that right? You can just make the agar put everywhere. What's a container for the agar? Long time ago, people use a rectangle one. Find it's lots of space. And you can do a math. You know the geometry. The same length, if you make a round one, the area will be much larger than the rectangle one or square one. So, agar at the beginning, about like 150 years ago, it's a rectangle. Right now, it's all circle. And they put in a tray. The tray is made by glass at the beginning. Right now, is made only also by plastic. This is called Petri dish. Why we call it a Petri dish? Because it's created by Richard Petri. And Richard Petri is also one of the group members from Bob Koch's research lab. So that's a story. There's a long story behind that. And this is Fanny Hesse. And later on, he has this idea. He also joined Bob Koch's group to do more research. He's also a great person in the microbiology area. Okay? Okay. Bob Koch developed the use of the agar to generate a medium, which is a bacteria solid media. What is that? What is the real components of agar? It's a polysaccharides coming from seaweed. So used to solidify nutrient bacteria and grow them. So most of the agar are polysaccharides. Bacteria cannot digest it, it remains solid, but bacteria can grow, and it usually will be melted at 50 degrees Celsius, and solidified at room temperature. That's the reason we can do pore plating, and we will do this in the lab. You will see it's very magic. It's melted, 55 degrees Celsius, we put in the water brush, and when you pour on the Petri dish, it's solidified about 10 minutes. This is the same thing like a pudding, like French pudding, if you like it or not. Something like that, the same material. This slide tells you using streak plating on an agar medium, get a bacteria pure culture. I usually do 3 or 4 cultures. This here is doing the 3 cultures. And this is a line use strict plating. And here you see the single colony, which means it's a pure culture, because it's only one genus name and one species name. This comes out in the last question. Binomial name system for bacteria. So something I would love to mention here, lots of people doesn't recognize there's some tricky behind this. Okay, what means a binomial system? Bacteria has two names. Let's say Bacillus subtilis. Let's say Listeria monocytogenes. That's called a binomial system. What are these names for? The first one is always the genus name. This is species name. Species. And they both have to do is italic. But there is one exception. It is Salmonella. For Salmonella, if you look at the journal or look at the textbook, let's say Salmonella Newport. Let's say Salmonella Thompson. The species name, you see, it's not italic. A genus name is italic. Why? Because Salmonella is more complicated. Salmonella, if it's full name, it is called subspecies enterica, subspecies, and Newport. These are italic. And people think about this name system is too long. Okay, what we do? I'm going to do this. I'm going to skip this. Therefore, when you see Salmonella, is always Salmonella Newport. The Newport is not italic. Why? The simple answer is, this is a subspecies name, not species name. This is a subspecies name. Okay? So there's one exception. This is for Salmonella. Others are look the same. Clostridium botulinum, E. coli, Escherichia coli, Streptococcus pyogenes. We will all talk about these in the lab. Okay? So this ends up with our first lecture. Exam 1, lecture 1, we talk about history, we talk about the pure culture, and all these information. We still have some time, so we're going to jump on to the chapter 2, lecture 2, exam 1, lecture 2. We talk about microscope and stain technology. This will be highly related to the lab, and there is a lot we could talk. First of all, is the microscope. What type of the microscope we have? What we are using in the lab, what we see it, it is called light microscope. Light microscope, there is a different category and different type of the light microscope. Why we call it a light microscope? Because the energy resource is light. Why we call it an electronized microscope? Because the energy resource is electron. That's different. So what type of the light microscope we have it? The first one is very simple. It's called bright field microscope, which is a common one we did in the lab. That means the specimen is colored. The background is bright, or you can say white. The second category is dark field microscope. Okay, the dark field microscope, which means the background is dark, is black, and the specimen which is showed up, white and bright. That's dark field microscope. Then you have a phase contrast microscope. Phase contrast microscope is because underneath the condensers there is a phase ring. Because the phase ring, the light going through the system, the wavelength will be changed. So you could do a direct observation. That's called a phase contrast microscope. The second one is fluorescence microscope. Fluorescence microscope is you using a different energy resource. It's still a light, but it's not a general light. It's a blue light or UV light. That is fluorescence microscope. The last one is laser confocal microscope. The laser confocal microscope is the laser is still a light, but it's a special light. And this one you could do 3D images. Okay, so these are the basic introduction about it. Now we had a light microscope, then obviously we have an electron microscope. Electron microscope has two categories. Number one, we call it a TEM, transmission electron microscope. Another one is SEM, scanning electron microscope. Transmission electron microscope we could see is the internal structure of a bacteria or a virus. Most of the time it's a virus. Scanning electron microscope, we will to see the cubic image of the surface. And the magnification, usually transmission electron microscope is much larger than scanning electron